Okay, going along with our discussion on fossils and fossilization and taphonomy, we're going to look at something we call paleoanthropology, which is essentially what we're going to be talking about for the rest of the course, or more importantly, what I'm going to be talking about, what you're going to be listening to, hopefully comfy at home in your PJs. In essence, paleoanthropology is the search for human origins. It's the investigation into our particular evolutionary history. So paleoanthropology, in a broad definition, is a branch of archaeology with a human focus which seeks to understand the early development of anatomically modern humans, right, through the reconstruction of evolutionary kinship lines within the family Hominidae, working from biological evidence, right? So in essence, this is a very fancy way of saying that, hey, we're going to look at all of our bipedal ancestors. We're going to look at everyone that has a similar skull shape and size to us humans, and we're going to see, is there a relationship between us and them, right? What's the relationship in terms of time and space? So the methodology of paleoanthropology is fairly straightforward, right? Of course, it begins with field work. You got to find an area that is full of fossils, right? With no fossils, there's no work. Then, of course, you have fossil extraction, which is a long and arduous process. As a matter of fact, some fossils can take years to extract out of the rock in which they're encased. And a good example is we're going to look at a creature called Sahelanthropus chadensis, right? When they found that particular individual, it was encased in very hard sandstone. And it took them 6 years of sitting every day with a very, very small automated needle that removed sand grains, grain by grain by grain. So you can imagine how daunting and time consuming of a process that could be. So once you're out in the field, you've got your fossil bed that you're working on. The question is really fossil analysis enough? Is that comparative zoology enough? Well, actually, it's not. Because essentially what you're trying to do is find a better snapshot or a better picture of how that creature lived in the past, right? So scientists may want a more complete picture, including the creature's environment. So a good environmental reconstruction can give clues as to what the fossilized creature's diet could have been, and more importantly, what their locomotion could have been, right? How they moved around their environment. DNA analysis can also illuminate evolutionary relationships between sister taxa in our taxonomic family tree. In terms of environmental reconstructions, they are, of course, based off of fossil evidence. So we're not just looking for fossilized creatures, we're also looking for fossilized plant materials, right? So we use local sediments to determine erosion patterns or whether or not a volcanic event occurred in the ancient past. There's also the use of meteorology, running simulations to determine what past weather patterns can be. And environmental reconstruction may also include palynology, right? You analyze that fossil pollen in order to determine what type of plants were living. So in this slide, you can see why palynology is so important when you're looking at ancient environmental reconstructions, right? Because a lot of plants, like our little pine tree that we see here, is a wind pollinator. You can see there are billions upon billions of pollen spores being released into the wind, right? And you can see in the picture here that pollen is fairly unique in terms of its outer morphology, right? So you can actually identify what pollen you're looking at based on its shape, the amount of dimples that it has on its surface, the amount of folds that are there, whether it's trilobed or bilobed, whether it looks like a wrinkled pea, or whether it looks like a spiky ball. You can use that morphology to identify it. Pollen itself is one of the strongest substances on Earth. There's a double phospholipid layer which allows twice the amount of carbon molecules. So essentially your pollen grains have multiple layers. You have an exine on the outside, which is an outer protective skin, and then you have an inner layer, right? And that double phospholipid layer on the outside allows for those carbon molecules to line up more precisely, which essentially creates basically nature or kind of the plant kingdom's version of carbon armor, essentially. So, and pollen is everywhere, right? All plants, all flowering plants produce pollen. So pollen can, in essence, be identified via DNA extractions, but that actually requires some plant remains to be present in order to identify it, and you can't get DNA from pollen itself. So pollen is generally identified shape, but that could be impossible sometimes. The example I like to use is two plants, one called Chenopodium and one called Amaranth, right? So we may all kind of know Amaranth. It's kind of an ancient grain that has been increasing in popularity again. And Chenopodium is kind of a backyard weed that we all have in Ohio that Native Americans used to use as a porridge. And essentially, what you can do when you look at Chenopodium and Amaranth, they look nearly identical on the outside. So what you do is you begin to measure, right? And by measuring the pollen, you can actually identify it more precisely when you have those shape discrepancies. So if we look at a case study, an environmental reconstruction of one of our ancient human ancestors, we have the reconstruction of the environment of Australopithecus afarensis, right? More importantly, we call her Lucy. This was done by Denise Sue at the Cleveland Museum of Natural History. So what she did is she obtained fossils, sediments, and pollen from an outcropping of rock in the Middle Awash region in Ethiopia. And that outcropping of rock dated to around 2 to 5 million years, right? So right during that time period that Lucy was existing in that landscape. So Dr. Sue's work was fundamentally extremely important in terms of how we determined Lucy's form of locomotion, right? So she determined that bipedality likely developed in a region which was still forested, right? The environment was pleasantly warm with an abundance of fruit-bearing trees and root vegetable plants. The forest was broken up by some savannas, but there was also an abundance of diversity in animal species, from small lemur-like creatures to large predatory cats and dogs. So in essence, what Sue's work found was she refuted what was long held in anthropology as the open savanna hypothesis, that humans were forced, or our ancient ancestors like our little Lucy were forced to walk on two legs because the forests were simply disappearing. And what Sue's work found was that simply was not the case, right? It was still very heavily forested. So there had to have been some other reason why we began the shift to bipedal locomotion. Some other methods of paleodietary investigation include, or paleoanthropological investigations include, paleodietary reconstructions, which is actually what my research specialty is. So you can reconstruct a creature's diet. This can give us clues to the nutritional availability within the environment of the organism. It can give us clues as to the overall quality of health that that organism had. And it can also give us clues as to migration patterns of that organism. There are several methods that exist. We have things like stomach content analysis. If you find an extraordinarily well-preserved specimen that still has materials in its stomach. You have dental wear pattern analysis. We look at what happens to the teeth over time, how are they worn down, and what plants and animals become more conducive to that process. We also look at, and more particularly what my research is dental calculus analysis. We look at the fossilized plaque that builds up on the teeth over time, and by dissolving that plaque in acid, we can actually pull out things like pollen grains, we can pull out muscle fibers and plant fibers, and can reconstruct the plants and animals that that individual was eating right before they died. So if we look at a case study in paleodietary reconstruction, we have the case of Leyuba, a 37,000-year-old mammoth that was found in the Yamal Peninsula in northern Siberia in 2007. She was so well preserved that the stomach, or the contents of her stomach, were perfectly preserved. So we know that she was eating various grasses as well as she was still milk feeding from her mother. We were even able to determine the cause of death, which was asphyxiation from mud clogging her airways. And when you see the picture of Leyuba, you're going to see this is one of the best examples we have, or one of the best specimen preservations that we've kind of ever found. If we look at another case study, we have one from a group of human relatives called the Neanderthals, and a long kind of held tenet in anthropology or paleoanthropology was that based on some of the nitrogen and carbon tests that we've done on Neanderthal remains, it showed that they were eating very, very high levels of meat. So we assume that these were individuals that basically had a diet that was exclusively meat, much like a large predatory cat or dog. But a study by Karen Hardy and Dolores Paperno, which looked at fossilized dental calculus, they found a whole mess of starch grains, right? So it really disproved the notion that Neanderthals only ate meat. And more importantly, it found that those starch grains were gelatinized, right? And gelatinized starch grains, they don't become gelatinized until they're introduced to heat and water. So this not only did this prove that Neanderthals were eating other materials than just strictly meat, it also proved that they were cooking their food as well. They used the specimens from a place called El Cedrón Cave in southern Spain. We're actually going to look at El Cedrón Cave a little bit later on in another recorded lecture. And it reconstructed a diet from around 50,000 years ago. So this is showing you one of the Neanderthal mandibles that Dolores Paperno and Karen Hardy used in their research study. And this is showing you kind of the gelatinized starch grain, right? We can identify where those starch grains came from or which plant they came from based on how they appear under polarized light, right? What kind of patterning do we see on the We also have another case study done by Christiana Warner from the University of Oklahoma at looking at dental calculus from 5,000-year-old human remains from the Middle East. She discovered specific milk proteins, right? And she said that the first milks came from cattle, sheep, and goats from this Fertile Crescent area. I will say, though, based on my own research, the evidence for this is kind of tenuous. Proteins themselves do not preserve in dental calculus, only their fossilized shells do. And we know that proteins, based on our talk about genetics, are very, very complicated 3D structures, right? So it's going to be very difficult to identify the specific protein just based on the outside shape. So let's run through real quick the kind of history of the discovery of ancient human specimens, right? And we're going to talk about all of these specimens in much more detail in our recorded lectures later. But if we're looking at the time period between 1840 through 1860, right, the first find was Homo neanderthalensis in 1848. It was found near the Rock of Gibraltar at Forbes Quarry. It was originally thought to be an inferior subspecies and not even human, right? Because remember, this is before Charles Darwin, right? This is before the theory of evolution by natural selection had even come into publication. In 1856, we have a Neanderthal skull cap that's found in the Neander Valley in Germany, right? This ends up becoming the type specimen for Homo neanderthalensis, right? And at this time, people believed that the differences between Neanderthals and modern humans were not based on an evolutionary difference, right? They were based on disease pathology. They thought that Neanderthals were simply a diseased subsection of humans. If we look at the time period of 1860 through 1890 and 1868, we have the first Cro-Magnon found in southern France. This was the first evidence of the antiquity of the human species, right? Because these were the ones that kind of first definitively looked more like us than anything that's been found before. And the first Homo erectus was found in 1890 by Eugene Du Bois, and this was the first quote unquote really definitively defined missing link. If we look at the time period of 1900 through 1920, we have a Homo heidelbergensis specimen found in Mauer, Germany in 1908. It's a new variant species on Homo neanderthalensis. It is actually a little bit older than Homo neanderthalensis. Discovery of the Australopithecus africanus skull in South Africa, the Tong child, found by Raymond Dart in 1924. And we have the Peking man, or as we call him today, the Zhao Cao Dian specimen in central China, but the specimen itself disappeared during World War II. So this is showing you in the left picture the Mauer jaw here. In the middle picture, we have the Tong child, right, which is a juvenile Australopithecus africanus specimen. And in the far right picture, we have one of the only known photos of the Peking man specimen. And finally, if we look at the time period of 1930 through 1960, the first Paranthropus robustus found. This is the first in a series of robust fossils that are found in South Africa. They originally named them Paranthropuses, but now we are comfortable in calling them Australopithecines, right? We have Paranthropus boisei found by the Leakeys in Olduvai Gorge in East Africa, right? This is the first specimen to be tested with potassium argon dating and dated to be about 1.8 million years old. We have Homo habilis being found in Olduvai Gorge by the Leakeys as well. This is the first human ancestor to be found with tools. So on the left photo here, you can see your Australopithecus robustus, or formerly known as your Paranthropus robustus. And here you can see in the middle, you have your Paranthropus or your Australopithecus boisei. And in the very right here, you can see this is your Homo habilis, right? And we'll talk in much more detail in the other recorded lectures about the specific morphological differences between all of these specimens, right? But on a very base level, we can see the one on the right seems to have a very, very large face, very big cheekbones, right? As well as the specimen in the middle. It has very large mandible, very big cheekbones. But as we get to Homo habilis, right, the kind of first member to make tools, we see that, the brain case gets a little taller, right? It's a little bit bigger, and the cheekbones aren't as wide and flaring, and the jaw structure is not as large as we see in the other two specimens.